Hello and welcome to the lecture Principles of Infectious Disease. This lecture is maybe the simplest one we have in this course because it is mostly terminology, and if you have crushed anatomy and physiology, by now you know how to memorize terminology. And some of this you definitely have seen before. It's always a challenge if in a class you're supposed to learn a word but you've seen it before. It's always a challenge to make sure that what you think in your head is the same as the definition that is technical and that you're supposed to learn. So I do urge you, especially if you see words you recognize, make sure you see the definitions and check them against your own definition. Okay, let's get into it. So this is the outline. If you want to look through it, you can pause. But what I've found historically with this lecture is that it's hard to keep track of what is what. So hopefully you'll be able to by looking at these slides. So a good question is, what words should you memorize? And the vocabulary list certainly has those words, but also in this slide, in this set of slides, I want you to look at the slide titles, because that's always a good idea. Is if a slide title is a word you don't recognize, that's an important word. But also some of the slides really are just terminology. So if the word terminology is in the title, learn the words in those slides. And then in a few other cases, I underlined the words you needed. So look for that. Okay, so the first thing we look at, disease versus infection. Disease is a bad state of health. It's a variation from the normal state. And well, you know what disease is. Infection is any situation where microorganisms are growing inside the body. And so this is not necessarily a bad thing. It's not always a disease-related thing. And that's maybe the tricky part for infection, is that it doesn't imply disease. And so what we're learning about in Unit 4 and what we're learning about today is infectious disease. And those are just diseases caused by infections. Okay, and there are words related to pathology, and you will see pathology used to mean the study of, the study of infected tissues or damaged tissues, or the ability to help diagnose something by looking at biopsies, and that's often referred to as pathology. But there's a more general term for pathology, a more general definition, which is the study of disease. And there are all these words related to it, and the most important one for us is pathogen, which I've used all along, and that's an organism that can cause disease. And we talk about pathogenesis when we talk about like what characteristics does it have that let it cause disease? Usually what proteins can it make that help it cause disease? That's how we talk about pathogenesis. And then pathogenicity is what we measure, like its ability to cause disease. So some more of this. This is a distinction we make between types of pathogens, where a frank pathogen is one that can sicken a healthy adult, but an opportunistic pathogen needs the person to be weakened or immunocompromised in some way. So the most common like opportunistic pathogen is usually Candida albicans is mentioned as an opportunistic pathogen because it's on us all the time, but it doesn't usually sicken healthy people. It sickens children and people with immunocompromised states, or people who have central lines, so they have a break in the skin that can be exploited. Also, the bacteria in your gut, many of them are opportunistic pathogens. So Bacteroides fragilis, if you've heard of that will attack you if it gets out of the gut, but it rarely gets out of the gut. And sometimes they work together in synergism. Koch's postulates are used to determine what causes a disease, like which microorganism causes a disease. Once you have a guess about that, you can do these observations and experiments. And if they all work out right, we say we satisfied Koch's postulates, we know what causes the disease. So let's look at what they are. There are 4 steps. The first one is whenever you see disease, you see that pathogen. So with COVID-19, whenever someone has COVID-19, you can find the SARS-CoV-2 virus in them. It has to be isolated and grown in pure culture. This is much easier with bacteria than it is with viruses, but you have to be able to grow it individually away from other species so that you can do research on just it, not a mixture of different species. And then if you put that into a healthy animal, you'll give it the disease. And then finally, that healthy animal, now that it is sickened, you should be able to recover the same organism from it because it should have grown inside that animal. So these are Koch's postulates, and they've been used historically a lot to help align research. When we can't satisfy Koch's postulates, we have to think of other ways to convince ourselves we know what causes a disease. And that can be a problem because we waste time studying the wrong things. One clear example of that is the bacterium Haemophilus influenzae. That bacterium in 1918, during the big influenza pandemic that killed a lot of people, they couldn't grow viruses in the laboratory then. They didn't know how to identify a virus back then. They didn't know what DNA was for. They didn't know things like that we take for granted now. So a lot of people would notice Haemophilus influenzae whenever there was an influenza case. It turns out it was just causing a secondary bacterial infection in people who were really sick because of the influenza virus. So the thing is, they would also notice cases where Haemophilus influenzae wasn't there. And so some people thought they had satisfied this first step, but other people thought they hadn't. And so that's an example of a situation where we wouldn't satisfy Koch's postulates because they just aren't true. Haemophilus influenzae doesn't cause influenza. It causes other diseases. So this is a slide that walks you through Koch's postulates kind of in a modern research setting, but it's the same, really the same steps. And then here are some exceptions to Koch's postulates. So if you can't grow the pathogen in the lab, you can't do this. So with viruses, historically, we couldn't ever do Koch's postulates. And here's an example, gonorrhea, the disease gonorrhea. We don't know much about that disease because there's no non-human animal that gets it. The only way we can do research on gonorrhea is by paying human volunteers. And so you can't do experiments on human volunteers that you could do on mice. But mice don't get it, so that's that. If there are a lot of different pathogens that cause the same disease, then you can't do Koch's postulates. So an example would be for the common cold. Something like 200 different viruses cause the common cold. Well, that first step of Koch's postulates wouldn't work. You don't always find a rhinovirus when someone has a cold. Sometimes you find an adenovirus or a coronavirus. There's a famous case where with leprosy or Hansen's disease, you can't do step 3. If you put the bacterium that causes it into a healthy host, into a healthy person, they won't get leprosy. Healthy people cannot get that disease. The only people who can get it are people who have an immune deficiency, a very specific immune deficiency, and then who are exposed to the bacteria that causes it. So just make sure you can think through a few of the exceptions to these postulates and make sure you know the 4 Koch's postulates. We will describe symptoms and signs. Symptoms are subjective, signs are objective, so you can measure a patient's signs, but you cannot measure a patient's symptoms other than asking them or observing them and guessing. So pain is a symptom, blood pressure is a sign, and that's that. Some diseases aren't well understood and we call them syndromes. That's where we have a collection of symptoms and signs that often are together. So many different patients have that same collection of symptoms and signs, but we don't know what causes it. So we don't know what causes it. But also it's used to describe the collection of signs and symptoms of a disease, even after we do understand the disease. So then we have diseases that are communicable or not communicable, and communicable diseases can be spread from person to person. And contagious diseases are easily spread from person to person. So there are a lot of diseases where the bacterium or the virus can spread from person to person, but it's not super fast or super easy. And then there are lots of others where they're very contagious. Measles is often given as an example of a very contagious disease. And Omicron variant of the SARS-CoV-2 virus. So the Omicron variant, that is a contagious disease. That one spreads almost as easily as measles, or equally to measles. But any of the strains of COVID-19 were caused by communicable viruses that spread from person to person. But that's not always the case. There are infectious diseases that are not spread from person to person. So tetanus is a good example, where the bacteria grow in you, but you're not going to give someone else tetanus. Botulism is the same way. These are all words that describe different infectious diseases. Acute versus chronic is an important thing to know. Clinical versus subclinical is important to know. And S/S means signs and symptoms. Latency, latent diseases, lots of us have them. If you've ever had chickenpox, you have latent disease because that virus is inside your dorsal root ganglia. That virus is there waiting to cause shingles later. One thing I've talked about when I brought up like Haemophilus influenza, well, we can describe that on this slide, where the primary infection was influenza and the secondary infection was the bacterium. And this often happens. Influenza is actually famous for weakening the immune system so that people get secondary bacterial infections in the lungs, whereas those healthy people, before they got influenza, they would not have been susceptible to that. So secondary infection happens after a primary infection has weakened a person's immune system. There's some very important terminology related to blood. So bacteremia means bacteria are present in the blood. Emia refers to presence in the blood. So bacteremia, fungemia, viremia, you'll see all those words. Bacteremia in particular is a weird one because it happens if you like floss your teeth. And so it's not necessarily a long-term, or it's not necessarily a disease state. And your spleen and your spleen, your lymph nodes, and your liver usually will clear out those bacteria. But that's different from sepsis. Sepsis is when you have bacteria actually growing in the bloodstream and the immune system is not effectively controlling it. And sepsis is surprisingly hard to diagnose. And so when we cover this in this class, I urge you to practice knowing how to spot sepsis. It's hard, and you'll find conflicting protocols for identifying sepsis. But it's a medical emergency because the person who has sepsis could face organ failure or severe shock that can be fatal in the near future. And so the sooner you can recognize sepsis in one of your patients, the more likely they are to survive. Okay, and that's enough for one video. That's plenty. This is terminology.